Much
this
better view.
looks different.
Shrimp hiding behind it too.
Any guesses as to
Yeah,
what that is?
Scott, what do you
guys?
think? Chris, Scott, I'm trying to count the little tentacles
Ship move
here.
is complete.
Reminds me of like Oculina, Ivory Bush. Yeah, go ahead, Chris.
This is in the family Plexauridae for sure. I think you can see there's very large. These are very large plates in between the polyps that are on the branches, which is one of the. It's sort of a modified sclerite, I guess, the way to think about it. So I'm pretty sure that's what it is. But going further than family, I'm not really sure. Maybe Scott has a better guess. Sacco thinks it's Parasus, and that's a really good guess too. If I was going to guess anything beyond family, I'd go with Parasus also.
Thanks, guys. Is there a reason for that sort of whitish color? Is that typical of that group? Or rather family?
Actually, they can have all types of colors, this particular group. They can be some of the most colorful gorgonians that are down there on the seafloor. In this particular one, the plates or the modified sclerites all along the stem there just don't have any pigment in them, and I guess there's not too much tissue. If there is, it's very transparent that's covering those plates, and that's why it appears white.
So is this a known species? Is this something we could identify down to species, or is this something new, never before been reported from this area, or new to science?
We're about to do a ship change.
Not really sure, but I think it's the same thing we might have collected on the first leg on one of these shallow dives. So I would sort of vote for, not bothering to collect the thing. Or even thinking about it and see if we can find something more interesting upslope, because I think we do have a specimen of it.
Okay, thank you, Chris.
Thanks, Chris.
Video, we can find that on the
So
serial
for
scan.
you on shore, your situational awareness, we have a pilot change going on right now. So just a couple people doing meal relief. And so we'll be up and moving
Video,
again once they are all situated.
On, the Sirius HD.
So maybe we can use this as an opportunity to talk about the mission. We are on the NOAA ship Okeanos Explorer, which is America's ship for ocean exploration, and we are coming to you live from a place called Supply Reef, and we're exploring unknown areas of the Marianas Trench National Marine Monument. So expeditions like this, the 2016 Deepwater Exploration of the Marianas, are only possible through a lot of successful partnerships and collaborations.
Ooh, Shirley, we have that
What are we
grouper
seeing?
again.
Where is it? Where is it?
Just over that ridge, there was another grouper. Sorry to interrupt.
No, that's okay.
Did anyone on shore see that?
There he is.
There it is. There it is.
Oh, it may not be the same one.
Zoom. Yeah, go for it.
Yeah, I was down at lunch when you saw the other one. That was
the
when you were
lasers
collecting
on.
the rock sample. You saw one? Yeah.
So
Oh,
Cruz-
wow. What do you
There's
think?
one off to the right. So I wonder if this is their territory, since there's
Bruce,
two.
do you know what kind of grouper this is?
This is Bruce.
Yep, go ahead, Bruce.
I'm a little confused about
There's another one
this.
further
It looks
down.
like it could be the same species. I think it is, but the color pattern looked different earlier. Now it looks more similar. It's got more of a white mark. Oh, there are two of them, by the way, in the area. And this is the kind of topography that they
There's
like,
another
where
one way
there's a
off in
little bit
the
more
distance. I
cover,
think
some
you may
large
have more than
leaves
two
to hang
down here.
around and orient to. But it looked like that one did have the bars on the body. It also has a darker
lose the
saddle
front, I
on
can
top
brighten
of
it
the
up out
tail,
there.
in front of the tail fin, on what they call the caudal peduncle.
Oh, there's another one. Oh, there are three of them.
Yeah.
Yeah.
Yeah,
Cool.
well, these groupers are well known for forming spawning aggregations, and I'm not saying that that's what we're seeing here, but you often see more than one in the same area. The aggregation for spawning also makes them very susceptible to overfishing, because fishermen will target those aggregations where they know that they can catch a lot of them in one area. So groupers are very susceptible to overfishing. Bob Humphreys, who's here and has worked a lot with it, says, yeah, they're definitely the 8-bar grouper, Hyperthortus octofasciatus.
Rotate
So I
left
will
and
type
maybe
that in
see a
here in a
different one.
moment.
So Bruce, you mentioned the coloration. So in shallow water groupers in the Caribbean, the groupers have the ability to sort of change their coloration a little bit. Does that happen here too?
I'm not
And in the Pacific?
distance. I'm not hearing a pilot.
I don't know for sure, but yes, they can, and I think it's highly likely that that could happen. There's behavioral control on the darkness, lightness of the fish, how the bars are. There could be changes with that color as they interact with each other, males and females, or changes during feeding and whatever. So yes, that's a very good possibility. We don't know much about the behavior of these deep water groupers at depth.
Getting
most
closer?
of them that are seen are ones caught by fishermen and hauled up onto the boat, and you don't see what they look like alive. So we were hoping for some bottom fish views for the interest of fisheries, and you definitely have that here with these grouper species. And again, because of the nature of their aggregation and the way that they form spawning aggregations, we don't know about that for this particular species, but certainly other groupers do that. They're seeing some observations of behavior in the natural habitat are of interest for ideas that might apply to management.
Thanks, Bruce. Yeah, I mean, one of our main partners on this expedition is NOAA Fisheries.
Yeah.
So finding this sort of information and being able to provide that back to the resource managers of the region is really great. Hey, Bruce, how many groupers do you need to be considered an aggregation?
I don't know if there's a precise answer to that. These are spawning aggregations. You're talking about dozens of them in a single site, or maybe even hundreds back when the populations were quite large. But that's a general term, a vague term. So they're not schooling fish. They don't move in unison with facing the same direction and swimming along. These groupers there'll just be several that kind of gather in a spot. So whether it's 3 or more is an aggregation is a little bit of a judgment call.
And do they spawn at certain times of the year?
Another look to the right.
Again, we don't know about this species. Very little information. I'll see if Bob has any ideas, although he's packing up and leaving. So
Which way are we going to be heading now?
Southeast, I
other
believe.
groupers, like the Nassau grouper, for example, in the Caribbean, yes, definitely have spawning seasons, and fishermen will learn when those seasons are. When fish accumulate in aggregations, and they would target those aggregations at particular times of year, which of course could have major impacts on the population, because if you're catching the fish as they spawn, they're not getting a chance to reproduce to the full extent that they might. And populations of things like the Nassau grouper, red groupers in the Caribbean were knocked down, depleted for that
probably going to
reason.
find that's a fairly short-term thing. Okay.
So Bruce, forgive me if you said this already. I don't recall you saying this, but there's something very, very fascinating about this genus and about a lot of the other groupers, in that they're sex-changing fishes. And if you already mentioned this, cut me off right now, but what we know about groupers is they're protogynous, sex-changing fishes, and what that means is first female. That's the Latin term for it. So they actually begin life all as females, and then at some point it can vary in different species, and perhaps with different social cues, they actually, some of the individuals turn into males. And it's a really fascinating process that I'm not sure a whole lot of people know actually happens in fishes.
That's really interesting, Chris.
This is Bruce again. That's the sequential hermaphroditism of first one sex and then another that I mentioned on a previous dive. When we saw the green eyes, they were synchronous hermaphrodites, male and female at the same time, but these groupers are sequential hermaphrodites. Bob Humphreys tells me from research that our group has done, and I don't do this myself, but at looking at the otoliths, the stones in the ears of these fishes that have rings, and you can also analyze isotope in these otoliths, that these groupers, this species, lives for at least 50 years. And since that sex change occurs with growth in size, the older ones that are one sex, it takes quite a long time for them to reach that age, 20, 30 years or more. Bob's gone now, so I don't have the exact age when the sex change occurs. And again, because they're such long-lived fishes, and because the sex change occurs at an older age, that also makes them susceptible to fishing pressure, because fishing will often catch the largest individuals, it'll be of one sex,
so hard to tell.
take those
which
out, and that affects the reproductive potential of the population.
So aside from the fishing and sort of man-made pressures,
sonar looks like
how
you're going
does
you're pointing the
that
right way.
sort of female
So
and
let's
then
call it
male
150.
change affect your
150?
general population dynamics?
Do you, Josh?
Sure.
Yep.
So you ready for a move?
Yeah, ready for a move.
Proposed distance?
That's right.
30 meters.
That's right.
Just following up on this, we were doing a lot of fishing surveys for this fishery for a number of years around the Hawaiian Islands, and we didn't catch too many of the Hyperthodus cornus, which we have in Hawaii, but it's very closely related to these fish. And we never caught a male of any of the fish that we caught. And part of the reason maybe is we never were able to catch any of the older, larger individuals.
30 meters.
Just sort of punctuating what Bruce mentioned, that it's quite possible the commercial fishermen are already impacting this species in the main Hawaiian Islands.
Thank you both. I learned a lot more about groupers today.
Boy, we're seeing a lot in the water column today, too.
Tara, zoom pink thing.
Okay, what do we have? Another... Is that another scorpion?
Another
scorpion, yeah, scorpion fish. And just up to the right of that, looks like another hexactinellid sponge. You'd see these spines from sea urchins along the bottom as well. Dead sea urchin spines.
Right. Lowers are
sponges one of the first colonizers of an area?
Lowers are in the
that's a good question. I guess it really depends on what's there. I mean, they certainly.
That was the light
for example, we didn't see a whole.
It looks a bit hot, right?
Did you see any yesterday?
Pretty close to the vehicle.
Maybe a couple sponges yesterday. Yeah, it really depends. It depends on where the nearest population is, how far the water meters travel.
I see that now. Yeah, maybe rotating them out.
Why are we seeing such a dense accumulation of this?
Okay,
really, it's probably one species with this demo sponge.
Going upslope.
Yeah,
so that's going to be
Why
hotter.
are we saying that? It might have to do with the way they reproduce. We know very little about that. We know they like it here.
Ship's about a third of the way through the move.
Okay.
So you can hear, we've been talking, for those of you at home, this mission, even though Patty and Casey and I are scientists on board who are leading the science team, this is very much a partnership among scientists, resource managers, really all over the world. And from time to time you hear some of our science team in land-based locations. Usually Patty and I and Casey comes and relieves us when we want to stop and get a bite to eat. And we do a lot of the talking, but we are also, it's kind of like phone a friend. I know sponges, Patty knows rocks, but there are a lot of other things out there about which we know very little, and we have a number of experts who are participating, who are collaborating on this mission, more than we'd be able to fit on this ship. So for me, it's really exciting because I get to work with a lot of people, scientists. Is that a fish right in the front, or is that just green rock? Yeah, I was looking closer. No, it's just green rock, I think. But I get to work with a lot of scientists with whom I would have never had the opportunity to work. And just
cool how
over the
these
last few
rocks
days, I've
just
learned so
laying
much.
right on
And
top
that's
of
one of the
the surface.
advantages of having a lot of people being
Clearly
able to
just, like,
participate in
dropped.
an expedition.
Did you guys already get zooms on these anemones further downslope?
The names on them, I'm not sure. Yeah, I believe we did.
Okay. Oh, and there's the yellow coral again. So yeah, it's one of the unique things about Okeanos Explorer is the way that we conduct science operations. And it's not just the scientists that we can have on the ship, but it's pretty much an unlimited scientific capital that we can have on shore. And we're just limited by people who have an internet connection. That's, at this point, an internet connection and a phone connection are the only things that are essentially the barriers between people and the deep sea, which is pretty fantastic. And to see how that's come so quickly over the last few years. When Okeanos Explorer first started, the only way that you could really communicate between the scientists and the ship was through an Exploration Command Center, or an ECC. And we used to have a special internet connection and a special phone connection, or it was via a intercom unit called an RTS. But recently we've been able to get rid of those barriers, and people can participate from their home institutions or really anywhere.
Yeah, and that's what's great is, for example, I participated in the first leg when Diva and Deb were out here. I was back in Florida, and it was quite late. And even though we have an Exploration Command Center at my institution at Harbor Branch at Florida Atlantic, it was late and late at night, and I participated in the mission mostly in the comfort of my own home, and very, very short lag time in the transmissions, and was really able to fully participate. So it's really a novel paradigm for expeditions, for exploration, for research.
The other thing that's great is that these Exploration Command Centers, so today you'll hear people from the Inoue Regional Center in Hawaii, from the University of Hawaii, from Harbor Branch in Florida, talking about, we're here at the ECC, and those places are now centers of collaboration where scientists can get together and work in a multidisciplinary group to work together to try and characterize an area, versus before that used to, like I said, it was the only way that you could talk to the ship, but you now have a much better connection no matter where you are.
And so in addition to physical scientists who are participating, biologists, geologists, we have a cognitive scientist who's at the Exploration Command Center in Hawaii. His name is Bill Clancy. And Bill, maybe you can, let me see, is he online? Yes, Bill, maybe you can tell us a little bit about what it is that you're doing. What's your role in this mission?
And below that is...
Okay. Yes, I'm at the Institute of Human and Machine Cognition, and as Shirley knows, I'm working with her and Debbie at Harbor Branch for over the last couple of years, and we've been trying to understand how the experience of being a scientist on this distributed team, working from home or in an exploration command center, how that's different from being at sea, maybe being in a trailer watching what's happening and controlling as part of a small group, or being in a submersible. And so I'm visiting the University of Hawaii ECC this week, learning a lot about our tools and how the work occurs, and I'm very happy to be here. Thank you.
Thanks, Bill. And it looks like we have something really interesting that came into our screen. Bruce, do you want to talk about that?
Well, this is another flatfish. It looks like another one of the left-eyed flounders. It's not the same species that we saw yesterday, that largemouth flounder. That one had a long fin ray on the front part of the dorsal fin. The dorsal fin is the fin along the back of most fishes. It is on the flatfish, but flatfish lay on one side. Both eyes are on the side that points up on the flatfish. That fin ray in some species is used as a lure. It's not bioluminescent. It doesn't produce as light. There's enough dim light at these depths maybe for other things to see, things dimly. People in submersibles can see. So, yeah, there you can see the fin ray. Actually, now that I'm looking at that, that fin ray may not be on the front end of the dorsal fin. It may be the pectoral fin sticking up, which makes gives me a different idea of what it might be. And I may have even had the family wrong on this. Boy, it'd be nice to get a closer view.
Yeah, we're going to try and do that. We're going to try and do that. Yeah, watch the pilot.
This could be in a different family, the Samara.
Yeah, if we can get a little bit closer and get a closer view of that, please.
Yeah, can you
Pectoral
stop the
fin
ship?
morphology, and some species are found in deep waters. But that's a real
Bridge
guess
staff?
at this point. I'll try to look and come up with a better
Could we bring
idea.
this move to a halt, please?
Anyway, those long fin rays are often used as lures to attract small shrimp or other crustaceans.
Ship
for food.
Thank you, Matt.
So, Bruce, the way that fish was moving, it seems very different than the way that a lot of other fish move. Would you mind explaining that for folks at home?
Is this a bossus, Bruce, with that large dorsal fin?
Yeah,
I'm looking at this, and it looks like the gills, the way the gills are positioned, this might be a right-eyed flounder with both eyes on the right side of the body. That would make this in the family Samaridae, Samaridae. I'll type that in. I think Samariscus. And now that I'm looking at it, it looks like it's got both a long ray on the dorsal fin, like I started to talk about, and this long pectoral ray that's sticking up like a sort of a flag on the top. Yeah, if you look at where the gills are on this, I can see that clearly. If you imagine the fish turns so that the gill opening's on the bottom of the fish, the eyes are on the right side, so this is not a bothid. This is not a left-eyed flounder. This is one of the right-eyed flounder species. As far as the movement, the swimming, it's very, very different than most fish. There are long fins along the top and the bottom of the body. Again, with the flounder lying on its side, it looks like they're on either side. And these fish creep along on the bottom using those fin rays, rippling them. We saw that with the tongue sole that was observed, I think, on the last leg, a couple of legs of the expedition ago, where the tongue soles were creeping along using these fin rays, moving them one after the other. They actually kind of walk along the bottom like a millipede or a centipede with their fin rays. Creep along kind of like a flatfish or a sea slug in the water, or a millipede or centipede on land. And he's certainly giving us a great view of that on the video with how these things literally walk along the bottom or creep along the bottom. So we're seeing this fish with the long fin rays, both on the dorsal fin in front of the that one fin ray sticking out in front of the eye and the mouth, and then the other one sticking up like a flag. I'll type in my guesses about this species. I'll have to do a little bit of research to find out what's been reported from the Marianas. It's a fascinating video, though, watching this animal move along. I've seen fish in this group. There's one species in shallow waters in Hawaii, Samariscus trialphilatus. Sometimes you can see diving if you look very carefully, and they look all the world like flatworms on the bottom when you first see them.
That's
right. They move and they creep along. And when I'd seen them, it's like, I look close. Oh no, that's not a flatworm. That's actually a fish. And this one's doing the same sort of thing.
I agree, Bruce.
Yeah.
I looked at it from a distance and thought, oh, there's a worm. There's a worm.
Yeah, I thought that too.
Polychaete.
It's a flatworm. I thought a flatworm or a polychaete.
That's probably one of the weirdest fish I've ever seen.
Yeah,
that was
pretty
so cool.
weird.
Thanks for the info, Bruce.
We're building our list of weird things we've seen. Thanks very much.
So you're behind me.
Yeah, thanks ROV team for getting us such great imagery of that.
Pick up and go ahead.
The ship is stopped.
Yeah, that's great.
Thanks, guys.
This has been a good fish dive.
So just another word about these flounders. Like a lot of the fish, they spawn eggs, very tiny eggs that drift up in the water. They have a larval stage. Flounder larvae, just like just all other kinds of fish, have one eye on each side of the head. As they grow and develop, as they approach the size at which they're going to drop out of the plankton, out of the water, one eye actually migrates across the top of the head, and as they settle down to the bottom, the eye on one side ends up on the same side as the other eye, and then the fish turns on its side, settling to the bottom, lives the rest of its life as a juvenile and an adult, moving on its side with both eyes on the side of the fish that's up off the bottom. Different flounders have eyes, both eyes on different sides. There are right-eyed flounders and left-eyed flounders. People used to separate the families of flatfish on that basis, whether they were right-eyed or left-eyed. But once people started looking at DNA and other types of characters, they realized the situation was more complicated, and that related species of left-eyed, right-eyed can be sort of mixed together in the same family, and the family divisions aren't as neatly defined by the eyes. There's even one species in the North Pacific, the starry flounder, that's found from Japan all the way over through to California. And I can't remember which way that it works, but on one side of the Pacific, the eyes are on one side of the head of the starry flounders. On the other side of the Pacific, the eyes are on the other side of the head of the starry flounders, and there's a transition where the proportion with the eyes on left or right changes across the north part of the Pacific and the Aleutian Islands in the same species.
Interesting. Thanks, Bruce.
And now we're seeing a sea star that looks, I don't remember, oh, and there's a shrimp over there too. I don't remember seeing the sea star before.
Looks different.
Yeah, it does look different.
It's interesting looking though.
Yep, yep, and beautiful shrimp.
Is it moving too slightly, or is that us drifting?
No, it's moving. It's moving.
I'm sure in a few minutes we'll have Chris Ma join us.
Yeah, I know. Chris Kelly says it might be Tamaria. Tamaria.
Video's clear. Pilot.
Yeah, you can come wide, just swapping.
So for those of you who are listening at home, you can tell we have quite a few collaborators, members of our science team, besides Patty Fryer from the University of Hawaii and myself from Florida Atlantic University, Harbor Branch Oceanographic Institute. We have scientists from all over the world. So you heard from Bill Clancy a little bit earlier. He's from the Institute for Human and Machine Cognition, which is actually part of the State University system of Florida. And I see, I'm not sure if she's on the phone line, but I see her in the chat room, Amy Bucko Taylor, who's also from Florida, from Florida State University. Asako Matsumoto is in the chat room. I don't hear her calling in, but Asako is a deep sea biologist, and she's from the Chiba Institute of Technology. So she's calling from or joining us from Japan. And another person who's on for quite a while and also in a impossible time zone to, I think I'm hoping she's sleeping right now. Is Tina Molotova from the P.P. Shirshov Institute in Moscow. Bruce Mundy is. Bruce, why don't you tell us where you're from?
Yeah, we were doing 130.
Okay.
150.
No, I'm a fishery biologist with NOAA, National Marine Fisheries Service here in Honolulu. I'm with the Pacific Islands Research Science Center, and I'm located at the NOAA
Next move
Regional
might be
Center.
145 to hit the waypoint.
So we hear a lot from Bruce. Bruce is our go-to person. He's our phone a friend every time we need to know something about fishes. Let's see, who else?
Okay.
Chris Kelly.
Pushing out.
Chris is from University of Hawaii also, and so is Diva Aman. Diva was the biology science lead on leg 1. Scott France, are you on the line? Scott's from Louisiana.
Actually, what was that?
And Tara Harmer Luke, I'm seeing from her little icon here in our chat room, it says I'm not here right now.
moving
But
around down
Tara's from Stockton
Saw like a
University
red
in
and white
New
stripe,
Jersey.
Something under that rock.
Who am I
Probably a
missing? Oh,
squat
Jeff
lobster.
Drazen. Hi, Jeff.
Squat
Are you on the
lobster,
phone
yeah.
line?
Just up in the right-hand corner near that white.
He is, but we're not keeping him anywhere near the telecon speaker.
Okay. And Mary Jo Browns, I think, is in Chicago today. I think that's what she said.
You want to snap zoom?
Mary Jo is actually at a lab at which she's doing some analyses, so she may have had to step out a bit.
Do a snap zoom?
She's multitasking today, and it's a good thing. She's had some wonderful commentary regarding the volcanology of this particular seamount for us.
I was just joking about Jeff. We have enough people in the UHECC now that he couldn't get a seat next to the microphone. That's why he didn't respond immediately.
Well, that's exciting.
Yeah, got to get a picture of that. Stick it up on our open house. Wow, that was a quick move. Little squat lobster.
And then what's that in the background?
Yeah, I was wondering what's right
I don't know
behind
if
this
that's
guy.
a bivalve back there.
It's kind of what it looks like to me too. Oh, this guy's coming forward.
So if any of you are having problems hearing us, please, let's see, I'm going to
just write something in the chat log.
Yes, actually do it in the private chat with Roland Bryan. He's in the chat room. So if you're having problems hearing us, please let Roland know. Just communicate with him directly in the chat log. In the chat room, sorry.
If we hear from you that we are breaking up, we will try to slow down our audio here by speaking more slowly. Right, Shirley? I don't know if Shirley can do that.
I doubt it. Okay, so we've had a question from the Inner Space Center, and someone there wants to know, what is the difference between a squat crab and a squat lobster, or are they the same thing? I have no idea. This is where I phone a friend.
Indeed.
And we both know from our early days of doing oceanography that a squat lobster is the same as a galatheid. That's what we always called galatheid crab.
Those are white, though, and the ones that we're seeing around here are this sort of orangey red. The galatheid crabs are just ubiquitous at hot springs, hot hydrothermal vents on the seafloor.
Yeah, so Inner Space Center sounds to me like they're the same.
Okay, we had a comment
Did you say 40 or
from
30?
Deb
30.
Glickson
30.
on shore, who is a geologist, about the nature of the broken up surface of the seafloor. You want to expound on that for us, Deb? What do you think this is?
I don't think that's what...
All right, 30 meters, 145.
Yeah.
Looks good.
Sorry, Josh, you said you saw an urchin somewhere?
Yeah, to your starboard.
Bridge nav.
Going.
Are you there, Deb?
We have a ship move, 30 meters,
I'm not sure
bearing
she's on
145 degrees.
not.
at
Okay.
0.2
I know she's online. I know she's in the chat room, but she's not on the phone line.
Thank you, Bridge.
Okay.
Okay, we see an urchin there. Do you have any theories about why this is all broken up?
Well, it's extremely thin pieces that we're seeing here. And my guess would be that the Mariana Arc is very seismically active. And I wouldn't be surprised if things get broken up, if there are earthquakes.
Oh, yeah.
Okay, can you come down a bit, co-pilot?
Copy that. Let me give you as much tether as I can right here. I heard we were underway.
Yeah, I mean,
I'll just keep a watch on it.
It's pretty flat here.
Sure.
Probably come down to like 13.
Yep, copy that.
Let's take Look at this here, if I can get out there. Yep. So that we had 30 meters going, 145.
145.
Now, Shirley, is this white thing another one of your delightful sponges?
Looks like on Sirius sonar, it's a little bit to port.
Okay.
Now I'm seeing fewer sponges here.
Yeah, I agree.
And see, there's more rubble, too. So
there's more, yeah. I think these are patches between the blocks of brown, broken up ash. I think we're seeing finer grain volcaniclastics. Well, certainly not finer grain than what is in the blocks, but still.
Okay, go ahead, video.
Lapilli to pebble-sized material. Yep,
Cold.
for sure. And that is a Gucci Samia.
With hydroids.
With hydroids. That's the fuzzy stuff around the base of
it is.
The stalk, right?
It is.
Are there any barnacles on this one?
Nope, I don't see any.
Do you have any idea why barnacles would like to be on the sea urchins
cool
more
rock
so
feature
than
up ahead
on
of
the
me.
corals?
Absolutely no
Is there a
idea.
reason we have
Maybe
this
it's because
Sirius camera
the
on the
urchins
left
move around more.
manipulator
Maybe.
monitor? Hey, Tara.
Total guess. I have no idea either.
Pilot, that was
You're
there when
looking
I
at
stepped in, so I'm
little
trying
tiny
not to
pieces
put away.
Switch that back
of
when you have a
rock here. And just as yesterday, where we were diving on a volcano that had erupted in 2014, there were some recently erupted fragments that had little white rectangular spots that you could see on the surface of some of the dark rocks. And those are tiny crystals of plagioclase feldspar that are very common in the rocks out here. And those are just crystals that begin to form in the magma as it starts, in the lava as it starts to cool on its route to the surface from down below. And it gets frozen into the rest of the liquid lava as it comes out on the seafloor.
Okay, let's zoom out, Tara, and then I can swim around while you're trying to figure that out.
Nice little picture of that coral group.
Pilot, I just zoomed your
And now
sonar
some boulders in the
Okay.
distance.
Find the
Ooh,
rocks.
big guys.
That's a nice one. Do you want me to
Ooh,
follow you
yes.
around there?
Let's take a look at the
How
big
much left in the
guys.
move?
And they have more
Looks like
opportunistic
8 meters
sponges
left in the
sitting up
move.
on the top.
Probably sponges, I would
Yeah,
imagine
you can turn it. You
corals.
can turn it a bit. It's
Yeah,
We're
it's
going to
flat.
find
You can
out
turn it a
in
bit.
just a minute.
Copy that.
So any
Are we going to be heading directly
idea,
east after this? Navi?
Scott
Yes, this actually
and
starts to
Diva
line us
and
up
Chris
right
and
better.
Jeff, any
Maybe we'll see where
idea
your sonar
why
sweeps.
we're not seeing
Yeah, even look at your
many
sonar. You're faced
octocorals
uphill now.
here?
Yes, sir. Looks like your swing arms are out good again.
Okay,
So flat.
so
Yeah, it's real
I'll
flat.
take a guess. For one thing, this is fairly shallow depths for octocorals, although we have seen them at other depths, in other places this shallow. But I suspect it has something to do with the substrate. I think this substrate is not really suitable for long-term attachment. It's broken up quite a bit. There are a lot of plates earlier. This looks a little more consolidated here. But
Hey, Tara, you
that's
can
my guess.
search for things at the top. I think
Thank you,
I think
Chris.
that would be monitor 6, I believe.
That is Chris
Yeah,
Kelly in
actually,
the
I
ECC
believe
at
it's
Hawaii.
monitor 2, according to
2?
my
Oh, okay. All
Actually,
right.
pilot, yeah,
Just have
that's
to-
fine.
find the...
So what we're seeing here looks to me like we have some urchins with the spines sticking out, some white sponges, and there's a patch of yellow encrusting
Yeah, it looks like
sponge
some yellow encrusting
on the
sponge
smaller
on that
rock,
one. Yep, over
right
to
in the middle,
Yep.
kind of just to the right of middle. And for those of you looking at this view at home, you can see 2 teeny little red dots right in the middle of the screen,
can
and
we
those
zoom,
are
Tara?
from the lasers on board the remotely operated vehicle. And that gives you scale.
there.
Those two dots are 10 centimeters apart, and that's about 4 inches.
Now those look like they are barnacles attached to the rock.
Yeah, rather than sponges.
Yep.
If you want lasers
The
up, let me know
really,
for a
really
shot or
small
anything.
white patches are sponges, but those larger ones are barnacles for sure.
Maybe we can zoom in a bit more.
Now, do you see a yellow incrustation on
over
the rocks
on the
as
right,
well?
Yeah, I do.
Do you have any idea what that is?
It's a sponge. No,
Is
are you
it-
talking
is
about
it-
the one to the lower left? or are you
Yeah, talking about that.
Yeah,
that bunch
probably.
Of sort of,
Yeah, you know what? It's probably a sponge
Oh,
as well.
wow.
These are notoriously difficult to identify.
Are they?
Yeah, well,
So I think
because they're so
we
hard
have to
to
go with
collect.
the analog image.
I would
on
guess.
that.
Yeah, you'd have to get the whole rock, and then I don't know how you'd get it off.
Yeah,
Okay, let's zoom out.
it's hard to do even scuba diving. So
So when you identify a sponge, I mean, I know that there are morphologic things in the sponges that help you to do that, but something like that that's so tiny, do you have to do chemical analysis, or what do you do?
Well, that's a good question. So most of it is microscopic. So we want to
Ship is
find out
Okay.
what kind of microscopic
Is that a
skeletal
good
elements it
direction
has.
for
They
you,
can be
co-pilot?
spicules. In
looks
shallow
like- I
water,
mean, I see that
there
rock
may be some that are
slided to,
calcareous,
port
and
there,
they
but
belong to a whole different
this is
class
good, as uphill as any
of the
in this
fauna
general
called
area.
calcarea. You never see those in the
I'm happy
deep sea.
with this direction if you want
And then for the demo sponges and the hexactinellids always have siliceous spicules. And
Yeah,
demo
it should be
sponges
roughly,
either have siliceous spicules or none at all.
roughly
So
east,
like
a
the
little bit
bath sponges
southeast.
we know, commercial
Yeah,
sponges,
right.
those are demo sponges,
We
but
could
they
start
don't
with
have silica spicules.
a little
Right.
bit northeast.
Otherwise,
It would
they wouldn't be-
scratch it to shreds.
Oh, right. It would be like embedding
I'm seeing kinda like
fiberglass
a-
into you every time you use them.
Maybe like a 100.
And so what we do is we try and study what kind of spicules there are.
You wanna do
Sometimes there are lots of
1, 0,
different
0?
types and how they're arranged.
Sure,
So the
yeah.
architecture.
Pretty
And
sure
we
that's
do that
just that
usually
one rock outcropping that
microscopically.
he's already been looking at. That's
Yeah.
on my sonar to the
For these
port
really
there.
thin
So,
ones,
yeah,
it makes it even more
happy.
difficult because
Turn
if
over
you
to
try and
100.
scrape it up, you can
It's so
damage
flat.
that
Yeah.
architecture.
Yeah.
So these are ones that are notoriously hard to identify
Kind of a choose your own
anyway.
direction,
And the spicules are arranged in different areas of the sponge. So you really need to have that sponge intact.
Okay. Tara, you've got the
Mary Jo
both
says
are
that the
on
lithified ash
port
layers look
manipulator
coarser
cams, so
grained
that monitor
here,
too will need to be,
and that the vugs are still
starboard
present.
manipulator.
There are occasionally some vesiculated blocks and coarse grained scoria. I learned that
Yes,
word
you
last
did.
week.
Bridge now.
And lapilli present.
Yep.
Yep.
What VCLs have volcanic
Starboard manipulator.
volcaniclastics.
Yes, ma'am.
30 meters,
What
bearing
vesiculated
100,
means for folks at home is it
0.2
just means
knots.
it has a lot of vesicles in it. And a vesicle is a frozen-in gas bubble. When the magma reaches closer to the surface as
fish.
it rises through the conduit of a volcano, it has gases dissolved in
Zoom
it.
in.
And we see a fish.
Yeah, another grouper. Yeah, when you were at lunch, Patty, we saw several of them.
Oh, gee,
Yeah,
I
I
missed
know
them.
you did. And Bruce gave a really good description of the groupers and how sometimes you see them in spawning aggregations.
Tunifor.
Yep, looks like a ctenophore.
Tunifor.
Ctenophore.
I think tunifor because it has two arms.
Maybe we could take a look at
Might be a phorophore.
Haha.
zoom in.
Yeah, we'll look at the ctenophore.
And as
I
we
mean,
zoom
the
in
coral.
here.
Oh, sorry, you wanted to look
Oh,
at the coral.
Okay, we'll do both. There's some
Do
little
both.
encrusting sponge things that are kind of
yeah,
blue-gray.
little patches of sponge.
And where was the coral? Where
It's
was
down-
the coral,
down below.
Chris,
Down
relative to where we are looking now?
Yeah, they have it. Pilots have it.
Might be a little too low, but I'll try to swing over. There it is.
There you go.
Beautiful. And this guy has a shell with, could be a hermit crab in it, or is that the actual snail? Maybe when we get a zoom.
So this is
zoom
the
in tighter.
other side of that coral that we saw a few minutes ago. As you can see, the polyps are mostly on one side, not on both sides. And we think it's a Plexaurid, family Plexauridae, and Osako Matsumoto suggested this could be in the genus Parassus, which I would agree with. And I don't know if Scott's on and get his opinion on it, but definitely a Plexaurid.
Got any more zoom, Tara?
Nope, that's full pilot.
Okay. All right.
That is a beautiful specimen of coral.
Yep. Shore-based scientists, we like this image.
And it's sitting on a piece of volcanic
zoom out.
plastic material, so those little black and brown and lighter gray fragments are all cemented together. Usually
that's great.
That cementing usually takes place as the material is deposited on the seafloor and is still warm, so they pieces of the material kind of weld together. So the ctenophore that I keep calling a tunaphore is sitting on top of some of the same material.
Okay, zoom in.
No doubt catching some particles.
Yeah, they like to be up high in the stream of water.
I have to tell you, until yesterday, I had never seen one of these. This is like amazing.
I've never seen one of them either.
Yesterday or day before yesterday, I can't remember the first time we saw them.
I think that was yesterday.
Yeah, yeah.
And yet they're fairly common here.
Oh, it appear to be so.
So I'm gonna muddy the water here.
Uh-oh.
We kind of sort of all decided that this is a ctenophore, but there isn't one single one of these where we've been able to see the filaments. And so I'm going to throw this out and see how many people bash it, that there is also a bilobed tunicate, I think, in some places here in the Pacific, and I'm wondering whether this could possibly be a tunicate, but I don't see any of the way it can breathe, the openings.
No, I don't either.
I'm starting to wonder whether or not this guy actually does put out its teeny or not, or whether it's on a nocturnal or diurnal schedule when it feeds, because something weird is going on. We should have seen the teeny on at least one of these guys by now.
Are we looking
Well,
for pilot?
yes, thanks, Chris. But, you know, yesterday what Scott told us was that these put their teeny out at night. So obviously we're not diving at night. I think I remember saying something like we ought to stay down here for a lot longer so we can see that.
A little bit in front of you.
Pretty sure he said that they come out at night.
I didn't hear that, so
I mean, you have time right now, though.
I didn't hear that, so excuse me. I take it all back. We're back to ctenophores, and you can just completely belay what I just said.
But no, I like this debate.
All right, let's zoom out.
What I was curious about when we had the close-up there was a little bump kind of on the back side of this thing that had like, I don't want to call them cilia, but they had little like hair-like things sticking out of the backside.
Oh, really? Oh.
Yeah. But what I understood from what Scott said yesterday is that the teeny are very long.
2 groupers there.
Is there a fish down? Oh, there's a bunch of fish. Those are groupers?
Yeah, more groupers, yeah.
Swimming around in
Maybe
the
the
crack.
same ones, yeah.
Oh, yeah, it could be.
Yeah.
Yeah, so this is Bruce. It's interesting that we're seeing these, like just small aggregations or just a few individuals of these groupers at these high relief blocky habitats. This is a sort of substrate sort of habitat that seems to be a focal point for groupers in a lot of areas.
That is not a grouper.
Is that a tuna?
That looks like a tuna.
Oh.
That's so cool.
You guys have a little bit of a delay on.
That's a dog-tooth tuna.
He's not going to cooperate.
Yeah, I can catch him.
Dog-tooth tunas are associated with islands that are not as open ocean pelagic as other tuna species. So divers see them frequently when they dive off outer slopes of islands.
Okay, thank you, Bruce.
Yeah,
and also thank you, Jeff. Jeff also put in the in the log dog-tooth tuna.
Sure. You were pretty far away from it, though.
Yeah. It's just when I start making maneuvers like that.
Copy that.
That's one thing you want to look out for.
Okay, we're going to do a nav check now. Shirley's going to go for it.
Watch lead nav.
Go ahead, watch lead.
can you give us some idea of where we are in relation to our next waypoint and how we're looking in terms of getting to all of our waypoints on this dive?
All right, well, we just left waypoint 3. We are 88 meters to waypoint 4, and then 170 from there to the end waypoint. And if we average,
I wonder if we could take a peek at some of those corals down there on the left if we had a chance. This looks like something we have an image before.
We have quite a lot of time. Yep.
Okay, pilot, I wonder, can we take a look at some of the corals on the left-hand side, if we could settle down a bit?
Do you have one in particular you want to see?
I think there's a crinoid at the base of the screen, just gone off the screen.
Any of those white ones?
One's in the bio box, port inner.
Okay.
Right.
I'll swing around here, set down.
There's a crinoid off to the very right-hand side of the screen, too.
Stalked crinoid.
Yep.
I'll look at the coral first, or the crinoid?
And that's one of the ones that has the...
What's easier for you?
It doesn't matter to me.
Okay, let's look at the coral first.
Yeah.
Let's get the coral.
So for anybody who's joined recently and isn't aware, the pilot of the ROV will position the ROV
go in.
So as to maximize capability of our videographer to zoom the large high definition camera on this individual.
So we have a 2 for here, crinoid and a coral.
So folks at home, why don't you describe this for us and for the video clips that will be available for people to look at.
This is a primnoid.
So what are we describing, the crinoid or the coral?
The coral.
So the coral's in the family Primnoidae, and the question is what genus? So there's at least 2 possibilities. One of them is in the genus Calagorgia, and the other possibility is something that's called Phanelia. And I can't really tell right now, but it's definitely a primnoid, and we'll just have to stay with that.
Thank you, Chris. And how about the crinoid that's waving at us? It's being very coquettish.
So I'm going to take a stab at this. I really don't know my crinoids, but that doesn't stop me to throw out IDs so far, so I might as well go ahead and do another one. This is, my guess would be something in the family Thalassometridae. If you look very carefully, right below that arm that's sticking closest to us, you can occasionally get a glimpse at his cirri, which is what he's holding onto the coral with. And I mentioned this the other day, that there are certain types of crinoids that have prehensile cirri. And this is an example of one of those where you can see the cirri that's wrapped around 360 degrees or more around the base or around the stem of that coral. And so that's what kind of leads me in the number of arms to something like Thalassometridae.
The pressure transducers, I could try that.
Yeah, because the one we saw yesterday that you were talking about, and you pointed the same thing out, was that it was one that did not have those prehensile cirri, right?
Doesn't really tell you much.
Right, and that's why I thought it might be a better fit for the family Antodonidae.
Okay.
What is that stalked item
Charles
that's
Messing and
right
some
near
others
the
are
crack
really the
up
experts
there?
on these things, and I've been trying to absorb every little nugget of information that Charles mentions in emails to us. But I really don't know that much about them. I just know a couple of characters to narrow it down to family.
Okay, so are all of these corals that we're seeing here the same thing? So that's question 1. And question 2, is it something new and something that we would need to sample?
So this particular one looks more like Calagorgia, but I'm not sure. I want to see if some of the other coral folks on the line, Scott and Asako, have another opinion about this and whether it's worth sampling.
watch leads are talking about this one here, or the previous
Yes,
one?
no, we're talking about this one.
Okay.
Right, and Asako just brought up a third genus, which I totally forgot about. She suggested it could be Plumerella, which I would agree. That's another candidate in the running for this thing.
Okay, let's zoom out and get a little closer, and I'll decide if they want to sample or not.
I'm not seeing any great calls to collect it, so maybe we'll pass on this.
Okay.
Okay, watch lead nav, no collection. Watch lead pilot, no collection.
Yeah, thanks, watch lead. Looks like uphill.
This is
Like
really
100.
irregular topography here.
Yeah, about 100 for me.
Yeah, these are pretty
Little
clearly
rain,
as
so,
we zoomed in on some of the
There
corals
wasn't
and
Should we get
the
another one in?
seafloor.
I'm
You
ready.
can see
Sure.
that the rock that we're
For
looking
100?
at is
Yes, it is.
layered, but it also is composed of a whole bunch of different sizes and colors of smaller volcanic clastics, and that just means broken up volcanic rock, broken up lava.
Bridge nav.
The eruptions that occur on these volcanoes are pretty explosive.
Can you guess what
And
we'd like?
so a volcano will throw out chunks of lava that are the
okay.
result of
We'd like a ship move 30
breakup of the
meters,
magma as it
bearing
rises in the
100
throat of the volcano.
at 0.2 knots.
As it gets closer to the surface, dissolved gases will come out of solution and expand within the liquid magma and just tear it apart. And so that's how you get these extremely explosive
Zoom in video.
volcanic eruptions around the Ring of Fire.
Hold there.
Okay. Now this is an interesting coral that has
this is different.
like 3 crinoids clinging to the top of it. The crinoids like
Okay,
to...
this is different.
All right, Chris, go ahead.
No, could be a. Well, actually, I don't know what it is.
Pilot, could we get a
This
close-up on that?
Like it might be more of a collection candidate than the previous one, but maybe
This
we can
is
get a
a
better
possible
look at this.
collection candidate.
Okay.
Yeah, go ahead, Chris.
I think Chris is
I'm
going to
just
wait.
saying maybe we could get
Yeah.
another look at this because this looks a little bit strange. I'm not sure I know what this is, but maybe somebody else does.
Okay, we're going in for a closer look.
Yeah,
Chris.
we asked the pilot if he could get us in position to take a real close look at this clump of coral and crinoids.
All right, the ship is on its way. It's gone about 5 meters so far.
Okay. Go ahead, video.
And here we're going in for a zoom.
Okay, just
Boy,
gonna hold
this
the
does look
establishment
different.
for a couple seconds.
The arms of this coral look more contorted than in the previous one. What do you think, Chris?
Scott's on too.
Scott and Chris, you guys are our coral
Oh, wow.
gurus.
This is different from what we've seen.
There's one open polyp in the bottom left.
Okay. Yeah.
You guys have an idea?
So Asako thinks it's a plexaurid.
Is that full zoom?
Plexaurid.
Yeah, she thinks it's a plexaurid. Scott had offered Gorgoniidae, but that was at a further zoomed out view. So I don't know if he's going to change the opinion or whether we have 2 possibles here, but maybe I can put it out. What does everybody think about grabbing a piece of this? Is there interest in this being a biologic collection?
Good question. Yeah. Just let us know. We are in position. And there are commensal crinoids up on the top of this guy.
I could stop the ship here, keep you guys comfortable.
Any opinion regarding
Well, my vote would be
sure.
yes. It's pretty unusual. I haven't seen it before. And Osako is suggesting it's in the genus Euplexaura, which I don't even know that genus. So
Wow.
I don't know. Maybe somebody else will weigh in on whether it's worth collecting.
Figure we'll wait till